Hello, and welcome to today's podcast. I'm your host, Stephen Fleming, and today we're interviewing my co-founder, Dr. Ryan Dilger, who is professor of animal sciences at University of Illinois at Urbana-Champaign. And Ryan was my graduate advisor, my principal investigator, back when we were researching this animal model. If you're listening in, I'm pointing to a pig on my wall. And Ryan has built probably, I don't think it's an argument to really say, one of the most, if not the most sophisticated facilities for raising and studying the behavior of piglets. We affectionately call this facility the Piglet Nutrition and Cognition Lab. Ryan is a man of many talents, but one of them is that when he wants to make something, he figures out how to do it and pretty much does it all on his own. He will source everything on his own. He will raise money to build it. Very useful talent to have in a graduate advisor. And today you get a glimpse into every conversation I would have with him in grad school, where we tried to stay on topic and failed. Couldn't do it to save our lives. But we do talk about, we worked a lot with industry sponsors in academia. And we talk a lot about building trust, performing interdisciplinary research, the value of data, precision livestock farming. I think we start getting into some hot topics, and then we kind of dance around those. But I'm really happy to be able to showcase him on the podcast, and you get to hear a slice of a co-founder chat. So without further ado, enjoy. Let's do an intro of who you are. Obviously, I know, but what's your educational background and how you got to being in your current position?
Yeah, so I actually started here on faculty in 2010 at the University of Illinois. I'm a full professor at this point, but have been only at two universities. So my bachelor's and master's were at Purdue University, where I really focused on non-ruminant nutrition, so working with pigs and chickens in a very applied fashion. So a lot with carbohydrates and enzymes and proteins and amino acids there. And it was really then wanting to work more with amino acids and get more into basic metabolism. Dive deeper is why I came to work with David Baker here at Illinois to do my PhD. So finished that in '07 and then started a 3-year postdoc with Rod Johnson. And that's where I got out of nutrition and much more into immunology, behavior, and that's then led to kind of the program that we have today.
Was that a conscious choice to say I'm going to do non-ruminant nutrition versus ruminant?
It was, absolutely. So I grew up on a pig farm, and there is a divide in nutrition that you are either with a simple stomached animal or one with a rumen. And we can learn a lot from both sides. And we'll likely talk about those different pieces as we think about, right, my lab studies the microbiota gut-brain axis, but there's a lot that we've learned from ruminant nutrition that we're now applying to companion animal to human nutrition because of bacteria, how important they are. But yes, it was a conscious effort because growing up, I wanted to be a veterinarian. That's what I knew who worked with animals. And it was going to be on the swine side of things. So poultry are a nice fit with that. We can learn a lot from those two species. But yeah, that's where my love's always been.
Why did you not continue with wanting to be a veterinarian? Maybe you still do want to be secretly.
I do not. I work a lot with veterinarians now, and there's certainly a place for them. But animal sciences is something that I didn't know what it was growing up. The veterinarian was the smartest person I knew who worked with animals. That's who I emulated. I didn't know what animal sciences was, or the fact that it's an animal scientist who's doing the nutrition, the genetics, and in many cases, the immunology and health that are right alongside individuals who have a DVM. So a PhD going that direction, that fits better with who I always was going to be. I shadowed a lot of veterinarians, did a lot of that type of work, and again, was pre-vet at Purdue. But as soon as I got into animal sciences, got into a research laboratory, that's where I knew I fit best. And so it's the organization, it's the drive, the scientific process. We all want more of it, but that's what drives me ultimately. And that's different than working in a clinical setting as a DVM, where they're not just fixing problems, but again, we can do things that work together between the animal health side and what we do on the nutrition side.
Based on what you said, like a lot of what interests you is a lot more research based. Maybe not to say that what you're doing isn't applied, but it is a lot more like fundamental science and curiosity and wanting to know how things work.
For sure. And basically every project that we do has some foundational fundamental piece that goes along with it. But if we can't tie it to a practical outcome with application to the industry in the next 3 to 5 years, right, we don't just want to do things that are applicable 20 years down the road. So yes, there is both a mix of kind of fundamental pieces, foundational pieces, basic science that goes along with the applied or practical science as well.
That's where so much of the research that I was doing in your lab on pig behavior was looking at all this literature that I think very few people cared about. It was like, how do pigs investigate their environment? How do they, do they or do they not remember objects they've seen before? Are they, aggressive? How do they learn? What do they know? And that was like where the split was, where I couldn't find these things in neuroscience or biomedical related journals, but there were a lot in like the animal welfare, animal science type journals, of course, just like using different names for the same thing. And even after I've left, I think those things have actually just become more important, because they're kind of just like, well, who cares, if they want to investigate toys or not? That's not going to like change the needle on production. But now you're a lot more involved in, okay, actually there is a tie between the neuroscience world and psychology and behavior and production.
Yeah, and that's the same challenge or opportunity that we have with our current students in my lab who are coming from a psychology or neuroscience background. Again, there's this entire wealth of literature out there that it is vernacular. We may use a different term for the same concept in animal welfare and animal well-being that from a psychological perspective we need to consider. And that's the difficulty, is you didn't necessarily have the background to review that literature or even find that literature, and it's where you watched me push you. But that's what I love about the program that I've developed here in our lab, and it really is taking that foundational nutrition ag background and marrying it with something that we can use the pig as a biomedical subject, and that's the species that does cross over between the two sides of the lab. Because I'm not just in animal sciences. I have affiliations at the Beckman Institute for Advanced Science and Technology, Institute for Genomic Biology, so at IGB, part of the Center for Digital Ag, AI Farms, National Center for Supercomputing Applications. All those things are not common when you think about swine nutrition. But we can use the pig in that way to understand things that directly apply both to swine industry, but also to the human health side as well.
I think that was the interdisciplinary nature of it was a struggle sometimes, because I felt this is a preamble to a leading question I'm going to ask you. It was like I have a foot in needing to understand human nutrition, but then also swine nutrition. And there's a foot in, because what I was doing in your lab was all related to pediatric nutrition. I needed to understand infant nutrition, but you have to understand in context of development. So it's human nutrition. And then you got to read the rodent literature because that's where, all the neuroscience stuff is done in terms of animals, or most of it. There were so many places where it's like, I feel like I don't know if fractured is the right word, but you kind of have to be a jack of all trades and then still try to be a master at the thing you're doing. And you have, affiliations all across campus, and you have this really, rounded out program. Do you feel that yourself, feeling like you have to be this jack of all trades, or have you been able to carve out this space where you're like, no, I'm definitely like mastering this aspect of all these?
So I don't intend to be a jack of all trades, but it is intentional to surround myself with diverse thoughts and bring in individuals who have diverse backgrounds and be inclusive of their path to that point and where they want to be able to go. My job is to help to develop well-rounded scientists and to know the bigger problem and how we can apply individual pieces and not be so siloed. The interdisciplinary research is something that is foundational to what my lab is and what I love, and it's why I can work across campus in lots of different realms. But I can understand what's happening in each of the different areas, but every student is going to be the specialist there. I mean, that's what your dissertation or your thesis is going to be based on, and that's why you have to defend that. I have to know enough to talk the talk in each of those different areas, but again, that allows me to help develop well-rounded scientists and make sure that when you get into the industry where you're not allowed to be siloed, you need to be able to. And I've seen many poultry nutritionists who have only ever worked on companion animals. or vice versa. You have to know, and it could get even stranger than that as you get into exotic species. But we can apply things, and it's the foundational knowledge. It's kind of our training that allows you to be that way, and to get that diversity in thought and diversity and problems you have to solve early on. I think it's important, and you're a product of that, to be able to say and talk in both the ag and the biomedical realms of things. So
first, you're definitely a jack of all trades in a good way, because you practically like built the whole facility that you do all the research in. Like, if that was me, I would have just been like, I don't know how to do this. I need to just like hire someone to do it for me. And you're like, nah, I'm just gonna just pretty much co-design it and build it myself, and like fix any piece that is broken. But that's been a big transition for me too, when going from the last years of graduate school and starting Traverse with you, and then running it, is definitely not being an expert at everything and having to I was going to say rely on others, but, like lead others, but bring in people who know more than I do, and kind of like relinquish this level of control. Because it's necessary, if you're the only one who, I guess if you're the smartest person in the room all the time, you're just, that sounds like, that's a really arrogant way to put it, actually. You know what I'm trying to say though, right? Like you're blind to what you don't know, and it's harder to grow if it is siloed.
Yeah, but I think it all comes back to building relationships, and therefore building trust and working with others in that way. And I'll be the first one to say what I don't know, because my desire is for all of us to gain more knowledge. And so I hope I don't come across as having that ego. I love doing science. It's what gets me up in the morning, what I get excited about. And the fact that my wife is also an animal scientist in meat science, right? That's all we do is talk science. And it's wonderful to see how we have some big problems that we have to fix. And I just came back from a meeting, an in-person meeting, where we're talking about climate change and how we can, in nutrition, in animal nutrition, help to be a part of the solution there, because we've been part of the solution all along. There are different challenges that we have nowadays, and we're in the midst of dealing with supply chain issues right now. Both in getting what we need to raise the animals and also getting the animals to become human food. But those are pieces that this is what we do. And we in animal, again, coming back to why animal sciences, this is how we are providing the cheapest, most abundant food supply based on animals on the planet. And we will continue to do that. But they're not easy challenges. It takes bright young minds like yourself to help kind of discuss this and get the story out there about why we're part of the solution and not just the problem, as you might hear it on social media. So.
We're going to quickly delve into all the big questions. Aren't you just killing the world? That's actually, that's a very poorly phrased question. But it is like, because of so many things being siloed. So before I got in your lab, I didn't even know that area of research existed, that there was like an intersection between, well, first off, I think I'd only touched a pig in like a petting zoo, maybe once or twice. Never even a piglet. And then learned of what you did. And then I was doing that for 5 years, and I'm like, duh, how does anyone not know that this is like, isn't this what everyone knows? And now that I'm out of that, I can come back and see like all the other things that are going on too. Everything is so siloed, and it's really tricky, because like I'd be with you and you would have this really eloquent explanation for why practices and standards are the way that they are in agriculture. Why is this the cheapest way to grow protein, essentially? And then I get out of that and it's just like, what was his explanation a little bit? And then you hear so much, and I think I've been almost more in the human nutrition side for a little bit. And, what's really popular is plant-based diets and foods. Which is like, it really does devolve really quickly into like an us versus them sort of mentality. But it's like, I almost like in the same day will read like, here's why plant-based diets are better for you, but then also see stuff about like animal protein and, a mix of why eating red meat is linked to cancer, but then also why it's like optimal protein, and it has the best amino acid composition. And actually, it doesn't do any of those things. There's so much information that it's difficult to like parse it out and Like, who's even right here? That's, yeah.
Yeah, so these are big issues that we're having these discussions with our students, and because they are these types of big issues, it requires you to not look at it from a single perspective. And the hard part is getting out of your comfort zone and accepting that there may be other perspectives, and even the same data may be interpreted in a different way. So the first thing we have to do is to accept that data are real, that facts matter, and then it's the interpretation. And that interpretation is based on our perspectives and backgrounds, and we all come at it with a bias, right? We work very hard in science to be unbiased and ensure that through the scientific process that we are getting at the right interpretation. But we all have a bias. We are all human. So it's this concept of, and we've always had politics and science, and we've always had other pieces, but social media is certainly pushing forward lots of more ideas, more diverse ideas, and not necessarily coming and checking our facts before we put them out there. So it is hard you can't control the narrative. And again, everybody's entitled to their opinion and their perspective in that piece. But we also have to be very real. And when it comes to food, the number one tenet, the thing that we always push, and when talking with students, is the concept that animal agriculture at its foundation is cleaning up things that humans do not want or cannot eat. And I would rather put those into an animal than to put them into a landfill, which is going to produce even more CO2 and methane and other carbon emissions. So the concept is that we are replete with protein in the United States. We are overeating the amount of protein that we need, but the rest of the world is not. And we're actually in a protein shortage the world over. So I like that we have lots of different ways to get there. I like that we can have diversity in plant proteins and bring other ways in. But it's not that we can't come at this foundationally that big ag is the problem. Big ag is providing what consumers want, and that's a cheap, affordable source of high quality protein in animals. And I think that there has to be diversity in the market. So if you don't want to eat meat, you are welcome not to eat meat. That segment is small and growing, but it's going to remain that way for a long time. And when you see the estimates when it comes to cultured meat, if we go that direction, get away from even plant-based meat, plant-based sources of protein, the estimates are that it may be 1% to 2% of the total market by 2050, and we're talking very small amounts, and it's not perfect in its own right. So I don't want to go all the way down that. That is what my wife does as a meat scientist and a muscle biologist, and Anna has. You should have her on this podcast sometime just to talk about where that's going, because she's right in the middle of that, and as part of the leadership in the American Meat Science Association and other groups, she's well poised to talk about those different pieces about why cultured meat isn't going to solve the problems of the world.
Yeah, she sent me a link to an article on that, and I kind of responded. I don't even remember. I remember one thing of what I responded. Like, part of me feels like, that's a common criticism of a lot of new technologies, right? Like, they don't scale well. They're really young. And thus, like, we don't need to worry about it, or they won't be successful. And this probably should just be a separate conversation after I've done more research. But it's like, well, that's like the argument against like solar power and other things too, that it's in its infancy and it's not energy efficient. But could be one day, right? Let's just stop there, actually, because I have a list of things I was going to ask you, and we like immediately dovetailed to other topics. I feel like it's going to end up like a Spider-Man meme of everyone just pointing at each other. You're wrong. You're wrong. I sent you a list. I think I'm actually going to ask you one of the last ones I was going to ask you, which is, so you're involved with the Center for Digital Agriculture at University of Illinois, and that involvement is related to, I think, a lot of the things that we were doing on the neuroscience side, where we were trying to, I think the questions we were asking was, how does, or at least I was asking in my dissertation, right, is how does pediatric nutrition impact brain development? What are the components in human milk, in sow milk, porcine milk, and in infant formula that need to be present for optimal brain development? And that required a lot of capturing video behavior of pigs and putting them in different arenas. And now, you and I talked about this a lot about, okay, well, what if that was to be adapted for pork production? And so this is another leading question for you. That seems so excessive. There's so much tech necessary to raise animals. Like, we can't just raise them and feed them. Like, why? This is the lowball question for you. Like, why does it require so much technology? Like, and you talk about how how you grew up on a swine farm, which certainly did not have the level of technology we do today, right?
Well, that's correct, but there's still a lot of engineering and automation that existed even on our small farm in southern Indiana. So the concept of having a curtain on the side of a barn that's going to raise and lower based on the temperature, that type of automation is one that doesn't require the herdsman to be there to maintain the environment for those animals. All we're talking about with the efforts that we have going on through the Center for Digital Ag and through AI Farms is really about labor optimization. And that's not much different than the producer didn't have to come in and crank the thing up. It automatically moved that curtain. It adjusted based on the ambient temperature outside. So the next evolution here is actually at the pig level and not at the barn level. And most of the engineering has been at the barn level to say that we have a motor that's out on a fan, that we have to change the temperature, that a feed line is broken. There was all those pieces in place, but that was at the level of the entire barn. What we're trying to get to is to the individual pig, and it really is about precision animal management and accurate delivery of what that animal needs in terms of nutrients and pharmaceutical care as well. So the concept here is all about computer vision. And so as part of AI Farms, I'm part of what's called Thrust 2, which is labor optimization, and basically we're working only in pigs at this point. And the concept is exactly what you and I were talking about when you were a PhD student here, that we can use that camera to identify what's going on with an individual pig, but it's beyond just the behavioral aspects, right? So what is the pig doing? Can that indicate that a health challenge is about to occur and give us more lead time than what the producer can see? So we're trying to get away from being reactionary and be proactive as to identifying specific behavioral traits or outcomes that are going to relate to health outcomes or otherwise. Quickly there, is a pig not eating? And if that's the case, can we move them to another pen where they don't have as much competition, or they have a different type of diet to help them to eat, so we can reduce the number of fallbacks and pigs that don't make it to market? But it's not just behavior either anymore. And we do a lot of looking at morphometrics of that pig, and we can identify that pig's weight just as accurately as that pig standing on a scale, using the length of the body, using a time of flight camera, a depth camera, to estimate its volume. So that's where working with Dr. Angela Green-Miller in Ag and Biological Engineering and Dr. Isabella Condata here in Animal Sciences, those are the other two animal scientists who are part of our team there. And Dr. Green-Miller is the lead of that thrust. But we also have all the computer scientists that are part of that as well. So we continue to have all the cameras at our Piglet Nutrition and Cognition Lab at Pencil, but we now have cameras on our commercial farms, our farms here at the university, and as well with commercial partners, where we're trying to identify individual animals within a group and that dynamic and how that works for identifying ways to, again, optimize the one or two individuals who are running that entire farm. We have the same issues with labor and animal agriculture, is that we can't get people to work on farms, and one individual is doing more and more. And most producers, at least in Illinois, have pigs, but they also have corn and soybeans. And so right now they're out in the fields and they're not walking the barns every day. You can imagine a time when basically the barn tells you there's something going wrong with this particular pen, take this action. So it allows the producer to be able to do more with less and to automate some of those pieces that instead of walking through, they're getting this information. And it's important to note that the only data we have on pig behavior is when a human is there to watch that behavior until we put a passive camera up. So we're just now, 2021, finally developing what is normal for pigs in terms of what do they do when there's not a person around, because we change their behavior in a group setting. So that's the type of data that we're producing right now.
A lot of progress has been made since I think we were both working on a similar challenge. That was like pre-COVID though. And then all that happened. How you have a really specific and unique setup for the environment you raise pigs for a biomedical setting, which is the Piglet Nutrition and Cognition Lab. Was it a replication of that in the commercial farms or the on-campus farms? Have you gone with a different method? How are you, capturing video and storing it and processing it?
So not a different method necessarily, except that we're now at Pencil, we're raising an individual pig in a space that is constrained. So they have all the space they need from an animal welfare perspective. But there's one pig on that camera and they have no competition for food, space, or otherwise. It's very different when you get into a group setting. And so we're still using computer vision as the main input that we have. And we're applying a lot of the same algorithms that we would use for tracking humans who would be in a crowd, right? But then we have to identify, again, what is typical. How much movement does an average pig do? And what is the threshold for under this amount of movement is something that we need to be concerned about. You've also got the group dynamics, so you can see where are the majority of pigs, and are they located in an area that's under a heater, therefore they may be cold or otherwise. And then you have the individual identification, and that is the hardest part. We're working on a lot of different pieces there, but identifying that it's the same pig over time and tracking it throughout that pen is very difficult because all the pigs look the same.
Yeah.
And so you can use things like RFID if we have ear tags, but not all producers are using ear tags in their pigs. We're working on facial recognition and other pieces. There's a lot of things that are very difficult when all the pigs look the same. They're all white, for example.
And it's not always like a optimal environment with high contrast and objects that do not overlap and retain in the same shape throughout, probably. Is it necessary to go to such a in-depth level to realize a return on investment for that technique?
That's the point. We don't know. We have to be able to identify at a high level and a low level which one is going to tell us the right amount of information to make an informed decision. And right now we don't know whether that's at the pen level or that's at the individual level for making those different pieces. But you can see different use cases or scenarios for each one. One may be more of this. The feeder, the single feeder for this entire pen has a problem. So all pigs are being affected versus this one pig is about to break with diarrhea or from an E. coli infection that we need to take some action on. So both of those we think are going to be important, but that's what we're trying to flesh out right now.
You touched on something that is really complex and elusive to me, which is what is typical behavior look like. And just like in ag, things are moving towards precision agriculture, and the human world is personalized nutrition. And I think that's in pet food as well. And we've both been part of projects where we're trying to look at data from, a decade of research, for example, of like looking at just your lab and trying to understand what is typical growth look like. And that was challenging for an honestly pretty easy variable. Body weight, pretty easy to capture, pretty easy to track. And on the human side, we have, guidelines for how you should eat based on what we think is a typical diet. And we're looking at these averages. And I guess what I'm trying to say is, how do you define typical? Is it based on the pigs in that pen? Is it based on that barn, all the animals in that barn? Then you're, managing multiple barns of animals. What's the relative, standard to compare something to, right? Do you need to know how, all behavior on average ever is? Or can you just say, okay, for this pen, this looks weird. Something's going on here.
It's hard to do it at the pen level, knowing that pigs tend to have personalities like humans. But at the pen level, even then, it's going to change over time. Remember, we're raising these pigs, these chickens, or otherwise over their productive life, but they're young. So they are also going through a period of development, in which case their behavior is going to change. So a lot of what is typical is based on what are the marketable outcomes, and that is the rate of gain of those animals, how much body weight are they accreting, and that's largely based on body protein because we are selecting animals and their genetics based on muscle accretion right? So lean tissue accretion there. It's the saleable products that are going to be used to the end. So a lot of that, what is typical, resides with the genetics company who says that when we've raised these pigs in this setting, here is what level of performance they were able to achieve. And again, in each of the different areas, but we are, as animal scientists, selecting those animals that are going to maximize efficiency and growth and therefore profitability, because we're ultimately selling a product in the end. We have to define what those standards are, what is typical when it comes to behavior, when it comes to other metrics that are not related directly to the product that the consumer is going to eat. So I think that's the fun part, is that we're in a wide open area of unknown at this point. And again, it seems like we should have made more progress before 2021, but that highlights the complexity of the problem that we're dealing with here. And I think computer vision is just one piece of the solution there. And I'm glad that we have such a wide network now of working with different groups, both in the industry and in academia and in government, to help solve these problems.
There's so many examples of that has surprised me, of like, oh, do we not know the answer to this question? It's 2021. And we've been doing a lot of lit reviews lately, and it's like when you drill down into the intersection of multiple things, you're like, oh, there's like 4 papers on this. Like, we actually hardly know anything. We kind of know a lot about each individual thing, but not how they all relate to each other. So it sounds like the first step is a lot of observation then. Just observing what happens under various conditions.
Absolutely. And there's also another piece there that we're very data forward, and we want to share the data. So we are creating data sets that can be used to benchmark and use for the development of algorithms, and sharing them in a way that others can benefit from that. And that is a relatively recent movement in science to be able to do so, to make sure that we're getting every bit of value out of that data set, because it is important. And we have those use cases, those scenarios are very important. That, again, we can raise animals, we can put a camera over a pen of pigs that's raised with optimal everything, nutrition, health, and genetics, everything that's part of that. But we also need to know what happens when health has been subverted, when management is not optimal. And we have some of those data sets that we're working with right now that would be very difficult to replicate, but we know that we're going to see this in a commercial situation. Yeah, and so we focus first on the pig, but also on the producer, because there are other pieces of this about lowly skilled labor or new labor who's coming in. How often are they walking through a pen? What is the human's behavior as they walk through that pen? Are there things that can be optimized there for the way that they interact with the animals or the observations that they're making? So again, it's great to have all these different challenges to be able to work on it, and to have the strong computer science group behind us as well.
Yeah, it'd be almost impossible to really replicate those things. Like, people talk about replication crises or crisis in science, but sometimes I'm like, look, obviously this stuff is hard to replicate. Like, it took $1 million to do this study. You're not just going to like randomly have 20 other papers replicate that finding, or even have 20 other studies that did the same thing. Stuff is just hard to do. And populations are really unique. You can't recreate the same population. How are you, approaching the data sharing problem? Is it, as simple as, let's, put our data in a GitHub repository, give people access to it? Is there something more user-friendly for people who aren't data savvy? But, why would you even care about people who aren't data savvy? They're not the ones who are touching the data. The great filter, if you can't download a repo, this isn't for you.
No, we have a number of Git repos as part of the different thrusts that we have, or the different efforts that we have, the sub-projects that are going on. But yeah, that is the main way of being able to share the data. But you've got to have an individual with enough technical skill to be able to process that data. And so data science is huge. And data science is a relatively new field. Every scientist thought that they were a data scientist until we put a label on a new field like this. But the visualization, and then going beyond that, is actually making educated management decisions based on that data. We have to be able to take that and put it in a way that every producer doesn't have to have a PhD in data science to understand what to do next. And that includes the algorithms for analyzing what the camera is seeing. and then how to put that together with other pieces that may not be computer vision, but taking kind of a holistic approach, if you will, to understanding what should be happening and what should the producer do. Help them to make the decision, which goes against my traditional upraising of the herdsman was the individual who was that computer, who collectively knew all that wisdom. But our producers in this country, the average age is close to 65, 68 of farmers. And we're not transferring that institutional memory like we really need to. So in some ways, computer vision is helping to supplement what's going on with fewer individuals. Again, that labor optimization.
You have a limited window of time to transfer the knowledge from those veterans into the algorithms to use, which I imagine is actually how you might train a lot of these things, right? Have an experienced person label the data, and then or not a lot of data, and then more after that, and more, and then try to see how accurate you can get your prediction to what that person would do. But then, as you said, like they see a really biased view of how animals behave, right? They by being present, they change their behavior, so you can get into entirely other problems. Although sometimes you do need the person there, right? Like you need to disturb the system in some way to see like how it reacts to I guess that's just one version of a stimulus. You can have a lot of different stimuli.
That's right. And there are, on the poultry side, especially when you may have 100,000 birds in one barn, there are robots that do that are actually going around and basically they're disrupting. Their birds have to walk, get up and move around. That's the time to be able to understand, do we have any issues with leg conformation? Are we having anything that we need to change with the diet? Because, you do need to disrupt at some point. But they're temporary and transient disruptions, right? They're simply getting up and moving around. But it's different in each context, and that's why we're focusing on the pig side of things. But there's folks who are working with poultry, with dairy, with beef, and others. So every one of them has different challenges. There's no one piece that's going to apply across the board. And again, that's the fun part. We have lots of work to do.
Yeah, it's in like every animal, insects, in aquaculture. Mm-hmm. This is a question I didn't actually send you before, but I think it ties more into your biomedical research, and like bringing different silos together. Off the top of your head, do you know of examples of how animal science and understanding pork production has impacted our understanding of human nutrition?
For sure. I mean, that's the pig, if not using the non-human primate, is the choice of animal model to use for looking at digestibility and availability of nutrients in humans. So a close colleague here at Illinois, Dr. Hans Stein, does exactly that, and using the cannulated pig, where we can actually collect a sample from the gastrointestinal tract to be able to identify, when feeding that pig human foods, how well is that being digested? That is a piece that we cannot do in humans ethically, to install a cannula into a human. Though it'll be noted that the first animal ever cannulated was a human.
What was his name? My grandpa loves telling me this story. It was a guy who got shot, and then he would put like meat on a string or something, and like put it in his was it his stomach or intestines? I can't remember.
Exactly. Yeah. So that's the concept, is that we do go back and forth between, and the pig is a wonderful animal model for many things in nutrition and immunology and behavior development, all the different things that we've been studying. It's the reason why. And the microbiota is one of the hardest ones, because the pig and the human are not necessarily the same when it comes to that, mainly because you hear on the human side how important butyrate is, and we don't have a lot of butyrate producing bacteria in the pig. We can take the pig, however, and we can humanize its microbiota, and Dr. Sharon Donovan, who's a close collaborator, does exactly that. So we can do those same types of studies in pigs that we can do in mice, and using a notobiotic pig and using a specific strain of inoculum to basically humanize the pig microbiota and do those pieces. So the technologies are certainly coming along that we can advance the science, but in many ways, if it works in the pig, it's going to work in the human. And that is different than hoping that what we learn in the mouse is going to translate all the way to the human. So the pig is a much more powerful and translatable animal model than when it comes to rodents. But we have to, again, acknowledge shortcomings. And in that way, some work with domestic pigs, as my lab does. Others work with miniature pigs and otherwise. But we have a lot of pigs at Illinois, and we are able to work with them in a way that is unique here. So both on the biomedical side and translating that, it's the fun part is going between the ag and the biomedical realms here.
Okay, I got to make a note here to come back to translation for a second. You mentioned butyrate, and what is butyrate? Even though like pigs might not have a lot of butyrate producing bacteria, commensal bacteria in their gut, butyrate is still used though as a feed additive, right?
Butyrate is used as a feed additive in both swine and poultry, and that's largely because we know that yes, it can stimulate the growth of colonocytes and be used as a primary energy source for cells along the gastrointestinal tract. So of course, that butyrate would largely be produced in the colon, the back end of the pig, by bacteria, but it's a volatile fatty acid, a VFA, or also known as a short chain fatty acid, and one that butyrate specifically has 4 carbons. So, right, you've got acetate, propionate, and butyrate, 2, 3, and 4 carbons, and so they are volatile because these fatty acids can go into the air. And butyrate in particular is quite pungent, right? You think of butyrate as rancid butter. It has a very strong smell. So it's on the animal side that we've developed the ways to encapsulate butyrate and therefore reduce its olfaction, the ability for it to have that smell. But we can use that to provide butyrate directly to the animal instead of providing a probiotic to seed that animal with the butyrate producing bacteria, or a prebiotic to stimulate the growth of those probiotics or those good bacteria. So yes, that is something that directly crosses over. And again, all of that information, what we know about butyrate, started in ruminant nutrition. VFAs, we put that vat of bacteria at the front end of a ruminant animal, and it's the bacteria there that are actually digesting the products that humans cannot and non-ruminant animals cannot use. So fiber, right? It's the fact that they are grazing on land that is not tillable, or they are consuming co-products from human foods that humans cannot eat because they are high fiber content and the wrong type of fiber for humans to want to use, but the bacteria can use them and therefore convert what would have been a waste product into meat and milk.
Yeah. I'm ambushing you by continuing down this butyrate chain. Do you know, is it specifically to deliver butyrate to the colon, or is it beneficial still to have that, in the small intestine and there's absorption there and it affects intestinal barrier integrity in the small intestine?
There are much lower, orders of magnitude lower concentrations of volatile fatty acids, including butyrate. And butyrate is going to be the lesser of those 3, so maybe up to 20% of the total, right, versus acetate and propionate. So usually that's the minority. Like colonocytes, there's evidence that yes, a portion of the energy requirement of that animal can be met through the use of butyrate in the small intestine. However, again, much lower concentrations, and by design, within the small intestine, we do not have nearly the numbers of bacteria.
Right.
So most of the fermentative capacities, either in the cecum or in the colon of a pig or a chicken, and we have, if we had bacterial overgrowth in the small intestine where we were getting butyrate produced, the negative effects of those bacteria would far outweigh the positive effects of butyrate being produced.
That would be bad, but I was thinking more in terms of like dietary intake of butyrate. That example of small intestinal bacterial overgrowth is a bad condition where you might have butyrate. But is it? I guess a better question is like, does it specifically have a beneficial impact on cells in the colon because it's unique to that environment, or is there a production benefit to feeding pigs butyrate, because that's going to travel through the entire gastrointestinal tract, right, and probably be available for absorption in the small intestine?
It is, and again, that absorption means we have a transporter for butyrate effectively, and with that, then you have to have those transporters being expressed in small intestinal cells and those enterocytes. But generally speaking, butyrate is being used by the gastrointestinal tract and not getting to the rest of the body, meaning it's used as an energy source by the enterocyte, by the colonocyte. And it's still a minority of the energy sources used therein. So we talk about first pass metabolism, and the concept is what we eat has to be digested and absorbed, but the enterocytes and the small intestine and the liver, they're going to take what they need, and predominantly they're using glutamine as a major energy source. They're using amino acids. So the amino acid pattern which makes it out of the liver for the rest of the body is different than what went in the mouth, right? And it's the same way with butyrate. That's going to be used by the enterocytes as a source of energy. But collectively, if you think about the volatile fatty acids, you may be 5 to 10% of the total energy that is needed of that animal. So it's still a minority versus then other sources using glucose, right? So breaking down starch to produce glucose as an energy source is going to be much higher concentrations there.
Yeah. You told me that, and then you allowed me to still explore back when I was on my VFA kick of, like, we observed this weird phenomenon, which I guess is not so weird anymore, where feeding pigs a fiber and prebiotic combination, this was polydextrose and glycoligosaccharide, improved memory of pigs in like the most cleanest, best, easy to interpret paper I've ever written. It's like never as simple again. And then I was trying to, a lot of theories on why the gut microbiome, or how the gut-brain axis works is on bacterial metabolites. And I was like, well, we should look at these VFAs. And you're like, they're going to be used up a lot in the colonocytes. I don't know. And I'm like, we need to look at this more and measure them everywhere. I don't even think we could recover them in blood or brain. You know, we looked at them all throughout the colon and then blood and brain, and we could only get tiny amounts of acetate and propionate out of blood and brain. And I think like most, I'm trying to think back at what actually made it to the brain, if it was acetate or propionate. But like one of them, it was like a couple samples did, and the concentration was extremely low. And that's where, like, when you talk about translation, it gets so tricky, because I would look at papers and it's like, oh, propionate induces autism-like behavior. And you go look at it and you're like, oh, well, you made a cranial window on this mouse and put propionate on his brain. Is this relevant to nutrition? I don't think so. Maybe in some sort of weird metabolic problem, there'd be a lot of propionate in the brain. But the translation part is tricky too, and you brought up, Sharon Donovan's lab, and I attempted to research choline in your lab, and then it ended up being all on oligosaccharides instead. And we'd get challenged sometimes because there's an argument that human milk is optimized for humans to eat. I guess, go figure. So why even research animals? Because there are gut bacteria in humans that will preferentially consume human milk oligosaccharides. Pigs don't have those same gut bacteria. So is it even relevant to, research animal models when you can't have like that same pairing? And I get you brought up notobiotic pigs. I'm rambling. I don't think I actually have a question here. I guess my question was going to be like, how do you, translate what's going on in the pig into the human? There's obviously a ton of rodent research, and there's so many examples of like, well, this worked really well in mice, and then it just didn't translate higher up. Maybe we're looking in the right place already. I feel like the work we were doing in your lab, maybe because it was informed by the human work, like we showed that it was beneficial in pigs to feed oligosaccharides, and then the human research is also replicating that. So like that worked nicely. I don't know if that was just coincidence. How do you translate what's going on in the pig to the human, I guess?
I think first it's important to realize that everything is relative, and that every animal model is not a perfect surrogate for the human. That the human is a different type of animal, has different drivers, different things that we don't have to consider in the pig. And one of those is, right, feed intake is something that we care about. How much does that animal eat? But that's largely driven by its physiological requirements in the pig. In humans, we're driven by a lot of other factors that don't deal with nutrients, marketing and otherwise. So we have to keep that into context. We have to keep in context how pigs eat. in that they tend to graze throughout the day and they're not necessarily meal eaters by definition. So there are pieces that there are pros and cons to every animal model, and that's not to downplay the incredible science that we've generated from rodents. They have an important place in science. But when you look at specifically in our case, the reason why we started working with the pig was the remarkable similarities between pigs and humans in the way that their brain develops, and even the structure of the brain, and then the function. So we look at learning and memory, simple tasks of learning and memory in very young pigs, because we can use human clinical MRI. We can look at structure of that brain to put that along with it. But we have to have animal models that are an important part of science, because we can do things that are much deeper. We can look and collect a sample from a pig that we could not from a human ethically, and be able to look at, again, the basis for why this may be happening. And you use the example of putting propionate directly into the brain. There's this whole concept that it may not be a humoral, it may not be passing through the blood, that signal is getting from the gut to the brain, that we're either through a neural input from the gut or otherwise. There may be other cells that are using that propionate within the gut that are then stimulating what's happening in the brain. But each of those studies is giving us a small, we have this large piece of knowledge we're trying to get, and we're chipping away around the corners. So we're doing so, again, using that scientific process to have a clear hypothesis, to define our objectives, and to have the right experimental design to be able to test that hypothesis. That's what's important. And once we can do so in a way that we know that the data are not confounded, then we can start to say, what are the pieces of this that would apply to humans? And we may have to acknowledge the shortcomings, what we can't do in humans, but how do we measure what is benefit, and translating that from one to the other? From my mind, it starts with using the same type of equipment, the same type of outcomes. And that's why the pig being bigger, meaning we can use a 3 Tesla Prisma MRI, is an important piece. So we're using the exact same clinical equipment for humans and pigs, and then we would expect that if we can overcome the smaller brain size, that pig has about a 10% of the size of the human brain. If we can overcome that and have higher resolution, but it's the same equipment, that's how we start to bring in that translational power. So it's just acknowledging the biology first, and that we have the ability to look deeper, get more mechanistic when we use the pig model, because we will never have the ability to easily design an experiment in humans to get at some of these very mechanistic pieces. So I'm not saying that it's all in hope and a prayer, but we want to have a model that as closely as possible replicates that of the human, and the pig is one of those.
Yeah, you're basically eliminating measurement error from they use this technique, but this group used another, which is always when you first start reading papers, the easiest thing to pick apart. Well, they didn't use the same technique. The pH was different in that buffer, or something like that. I guess you're kind of removing the species-specific element of it, and that was why it was difficult, I think, for us to ever get to the point of measuring things like anxiety in the pig or resilience to stress, because the way those are measured in in rodents is very species specific, right? They freeze, or they avoid open areas. If they develop a fear response to something, they exhibit freezing behavior, and there might be elements of that a pig exhibits too, but nowhere near the extent that rodents do, making it really hard to say. A lot of work on prebiotics and fiber in rodents shows that it reduces depression-like behavior and anxiety-like behavior. And it's I can't even reliably measure that in the pig. So it was kind of we tried, and either because the method didn't fit or because the ingredients don't actually act that way, it didn't affect anxiety. So I guess you do have to just pick wisely, the sort of subdomain that you're going to look at. And
but again, that's something I'll say yet. We haven't identified how best to study anxiety in the pig yet, but that's an area of active research, such that, again, the benefits there are also towards animal agriculture in reducing any type of stress that we have in that pig's environment, in the same way that inducing anxiety and understanding how a nutritional intervention is going to hopefully negatively influence that stressor will apply to humans. So we have to know enough about our animal model and what's going on in a species that can't communicate. right?
Yeah.
They can only express their natural behavior, and we be able to interpret what that means. But we have to do the foundational science to understand and make sure that we are interpreting that correctly, designing that type of model and that type of outcome.
It's very like I'll know it when I see it, but I can't actually put anything forth to define it.
Right.
I know when Henry is upset, but he's not telling me. He's just screaming and yelling, and a pig or a mouse might do that too. But like that was so difficult though, coming up with like the behavioral ethogram and like, well, okay, well this pig seems to like jump around a lot. Is he playing or is he trying to escape from the arena? The context was so critical. And the same exact thing would, I guess maybe obviously, but the same exact stimuli can induce very different responses, which then just makes statistical design really tricky, because you're going to like not have near the power that you thought you'd have.
And we know we have variability, especially in behavioral outcomes. Again, the beauty of the pig is that we have close control over the genetics and the background, and such that with a litter-bearing species, at least there, we can have siblings represented on every treatment, and we can do both sexes in the same way. So we have to take those considerations into play, where we know that type of variability is much more amplified when we get into humans. So it's about defining, having a clean model when it comes to the pig, and then understanding that it's going to be even a more variable response in the human. That's an acknowledgement we have to make from a science perspective.
I suppose it could be viewed as, well, no, it definitely is a challenge, but that's kind of where the precision aspect comes in, right? And on the human side, all the personalization comes in, is through all that variability. That's how you're going to make a decision to use a different production technique maybe for a given pen or barn, or feed someone, a different type of diet. But then, you just have such a huge initial obstacle though, to get enough data to figure out what the right response and stimulus to provide is to maybe reach the same outcomes.
So that's why I actually prefer the term personalized nutrition.
It's so funny. Some people like precision way more than personal. There's like, I don't know, but yeah.
But we want it to be both accurate and precise.
Yeah.
And that's what personalized in my mind kind of brings that together. But I was having this discussion with the group just over the weekend of, we want to be very precise, but we also want to make sure they're getting the exact concentration of nutrients, right? So the accurate side of that coin as well.
Very precise in the wrong ways. Accuracy, right, is like on average you're hitting the center of the bullseye, and precision is the basically the variance, low variance.
Exactly, and we want both the hit the center of the bullseye, the correct concentration, the correct everything, but also then that everybody is hitting the bullseye, and not that we have this large spread in variability.
Yeah. Means are not sufficient.
And you can say that personalized means that you are talking about an individual and ignoring the variability.
Right.
But I think that it's getting down to the individual level, I think, is what's going to be important there.
Yeah.
And that, again, with animals in a group setting, just as it would be in an individual setting or a human as well.
I think, and I think the data sharing that you're doing becomes so much more important, because out of publications, you're just getting means, and you're just getting averages and some measure of error. It's not always reported if it was even standard deviation or standard error. You're so limited in terms of what you can get at. And it's actually kind of frustrating, because you'll see papers where like they actually could answer my question, but they weren't looking at it in the same way. But you can email them and ask for access to data. There's just too much like friction to get to the point of like, maybe they're not fun to work with. Or they don't share well, or anything like that. And I was at a conference a little while ago, and this was more about personalized nutrition. And this group had run a really big trial, providing personalized nutrition advice, whatever that was. And someone said, well, now that you have this huge data set and it's personalized to everyone, can you now come up with a recommendation on, what we should be eating? And they're like, absolutely not, because that recommendation was awful for X percent of our population and really beneficial for another. And I was just like, oh my gosh, we don't know anything. I'm lost now. Oh man. But you need to be able to at least I guess what I'm saying is it's not going to be possible to rely on like averages anymore. I guess it is averages, but you'd have to just drill down by like every category and context and interaction between multiple variables and say, okay, well, for this subpopulation of a subpopulation of a subpopulation.
Right. But I think, again, our challenge is easier met on the animal side, where we are delivering a specific diet to a pen.
Yeah.
And therefore we're feeding to the average of that pen, knowing that there's a curve, a normal distribution within that pen, but at least that's their only source of nutrients. And that's, again, where we have a lot less variability there, because we've controlled the genetics, the environment, and the diet of that animal. Whereas on the human side, we're eating whatever we want. And we're not eating the same time, the same nutrients. We have a much more diverse diet than we have when it comes to animal ag.
I think they're both pros, but in very different ways. Because, well, I was going to say that's a benefit of humans in some ways, but that's provided you have a sufficiently large data set to say that, like, okay, we did have a lot of variety or variability, but we still had enough power to say for these people, we can investigate some sort of statistical question. But when you have like a sample size of 20 or 30. That's out the door, and you need to have something that's more controlled. So we talked a little bit about what you're doing on kind of like the AI farm side and Center for Digital Ag, but on the nutrition and biomedical side, well, first off, a lot of the work that you and I have both done is sponsored, so we can only talk about it after it's been published. So I actually don't know how much you can talk about, but what direction has it gone on the biomedical side?
Yeah, so things, I mean, we can talk about kind of my academic interests in those different ways, and largely the work that we do with sponsors is kind of feeding into those. So they have some similar questions, and you've touched upon choline. We've worked a lot with iron, as well as what are the important factors early in life that are going to have an influence on the microbiota, the gut, and the brain ultimately. What we're doing now is we're starting to lengthen that out, and most of our studies had been only 4 weeks long, so early in development, where we're now taking them out to 8, 12, 24 weeks of age, where we're getting into a sexually mature pig, and therefore going across a much larger span of brain development. So some of it is just against what is typical for brain development of the pig, and we've developed the Brain Atlas, working with other groups outside to improve that Brain Atlas. We know over time how those curves are being developed in terms of mainly structure, but also some composition and thinking about myelin and otherwise. All of these things go together because we can sit at this place, use the pig, and we have these public-private partnerships. So both funding through the USDA, through NIH, NSF, but also private companies, we're all heading in the same direction. And it's important that we have that diversity in the funding model to ensure that we can keep asking these tough questions. But it still comes back to the individual students. And I have usually 1 or 2 students who are focused on neuroimaging, 1 or 2 who are on the psychology of the pig, if you will, so the well-being and behavioral aspects. But we continue to kind of broaden that team out to ensure that we are asking those questions appropriately, designing the experiments appropriately to make sure that if we are studying anxiety, and that's a push that we have right now, are we interpreting that correctly? What does an anxious pig look like? And is that the same type of anxiety that we would see in a human? So making sure that we are thinking about those pieces critically when expanding the model. But we continue both thrusts, both the behavioral side and the neuroimaging side. On the neuroimaging, we're very blessed to have the Beckman Institute here. Paul Audibert celebrating the 50th anniversary of the first MRI, and that's on display here at Illinois right now. But the concept that we're not only using a 3 Tesla magnet, which is a standard clinical piece of equipment, we're now working at Carle Hospital with a 7 Tesla magnet that is what humans lay in as well, but giving us even higher. We have the ability to get our pigs into the hospital, and then we have a 9 Tesla magnet here as well for doing younger pigs. So we continue in these different areas and thinking about, again, structure and function and those relationships as they're being influenced by nutrition. And in many ways, we can look at both positive and negative nutrition. We can understand if we don't have enough of a critical nutrient, how does that influence things in order to improve the way that we're feeding humans, in that case, babies. So ensuring that choline is there, the right amount and type of iron is going to be present there. And as well, we can look at probiotics and prebiotics to understand if we're influencing the microbiota early in life, is that having positive benefits as well? So all of these are what we call technologies, and we need to continue to develop those technologies and understand individualized nutrients and entire dietary patterns as being good or bad, and how they're influencing the biology ultimately. But we basically are taking cues for what is important from a human perspective and what are the clinical outcomes that are possible. And that's why we are now comparing pig brain development to human brain development. Again, getting back to that translational piece, what are the similarities between the two? How do we look at a chronological age between them and say, this is when the hippocampus is fully formed from a structural perspective? Those are things that you've been a part of as well, but are important for defining the model that we're working with.
Yeah, and so okay, you're kind of along the same track, but you're doing much longer studies in longitudinal, which is nice so that you don't always have the conclusion, which is like, well, longer studies should be done. Less cross-sectional. That's always, it's like you read between the lines of, like, you could publish so many 4-week studies, but it obviously just takes longer to do a 24-week study. But, like, it still might be one publication.
But in the end, if everything we did in early life meant that every time they all had the same brain, then we're not actually affecting overall brain development.
Okay,
so that's why.
Yeah. This is what I was going to ask you, which is, surely the answer is it depends and varies, but at 4 weeks old, we would sometimes say is kind of like a 4-week-old piglet is kind of like a 1-month-old baby if you went just on volume, which is, super crude measure. The question was always like, okay, this thing went up or down. In the most unscientific sense, we would go, okay, is this a good thing or is this a bad thing? And there's on the brain side. By the way, I know you're going to have to go to another meeting soon, so we'll make this like the last question. It would be like, well, we could argue that a bigger brain region is better because surely more brain is good. Or the reverse and say, it's more economic in its function, so it doesn't need to be as large to achieve the same function. Do you think does it matter if there's like a perturbation or change early on if we all get to the same place at the end? Are some of these things just like transient and it's just kind of like maybe thankfully they're not as important as we thought?
I mean, that's fundamentally what we're trying to study. We're trying to understand that if we did have a perturbation but we still got to the same point, then that perturbation either didn't matter, or if at that same point we had a different stressor and they responded differently, then we may have a quiescent, something is quiet. We've changed the way that these cells are working. And in many cases, we can only see what is not normal by subverting the system in some way. And we use immunological challenges often for that in my lab, but we may use anxiety or others to understand, do they have a better response there? So it's and you misspoke before when you said a 4-week-old pig is equivalent to a 1-month-old human. It's more like a 1-year-old human.
Oh, okay.
So, right. So 4 weeks is 1 month. Mm-hmm. They're not equivalent. So that pig is going to be sexually mature within 6 months, whereas we're talking 13 years in the human. So it is that's a nice part about the pig, is that brain development is much compressed relative to its overall lifespan. But the concept there is that we can study those pieces in the pig and be able to apply them to the human after some amount of time. But they are difficult questions to ask, and we think more about the developmental rate, and we still have to figure out how to interpret that. If we enhance or if we increase the rate of development, is that a good thing or a bad thing? And I think it depends. We don't know yet. Yeah. And volume is one piece of that, but of course it's a dynamic process, right? The brain is plastic for a reason, and we are going to reorganize over time. Structure is just one component of that.
I think we've seen it in multiple ways where we might see an improvement to memory early on, but for example, when in some of the iron deficiency work, we saw no effect on memory, and then after they had gone on to an iron replete diet, we saw some sort of impairment, which was, odd. It's kind of like this latent, as you mentioned, quiescent. Is that a real word? Quiescent. I'm going to look that up in a second. You're looking at me like you should know this, you biologist. Come on. Just haven't used it in a little while.
Yeah, but latent, right? That
Yeah.
it wasn't immediate at the time. They all looked the same, but then it was only after you'd gone through other developmental periods that they didn't respond in the same way. And that was an additional 4 weeks of being on an iron replete or iron adequate diet. So it was the early life iron deficiency caused later in life changes
Yeah.
in structure and function of the brain.
See, this is just my bias coming in because, researching infant formula and human milk for so long, then we have a baby, and then he's needing formula in addition to breast milk, and you're just going like, oh, but every introduction to these papers says breast milk is best, and now I'm not doing what's best. So I'm just, trying to get at like, hey, hopefully it'll all be fine in the end. Hopefully the lines converge on the top right part of the graph, or wherever things go.
Yep.
Okay, well, this has been good to have you on, and ironically, the co-founder to this company wasn't the first podcast episode, but that's okay. We still got there, and I'm sure there'll be more. So thanks for coming on, and I think we brought up a lot more questions than we answered, but we'll continue to pursue those.
No, and I appreciate the opportunity, Stephen. Many of my colleagues only know me in one area. I'm either a poultry nutritionist, a swine nutritionist, or I'm on the biomedical side. So it's good to be able to talk between these and then kind of bring all the different areas together. So thank you.
Yeah, absolutely. All right, take care.
All right, thanks, Stephen.
Hey, thank you so much for tuning in to the Traverse Science podcast. If you'd like more of this content, check out our website on traversescience.com. You can follow us on LinkedIn, subscribe to us on YouTube, and you can check out the podcast audio version on Spotify, Apple Podcasts, and Google Podcasts. All right, take care.